Is the genetic factor. We will talk about genetics, and we have two sections. In section 1 we will talk about introduction to genetic and about Gregor Mendel, his observation, results, conclusion of his experiment. And in section 2 we'll go to talk in detail about the crosses of monohybrid, dihybrid, test cross, and the others. Now let's start with section 1. Here you have a history of Gregor Mendel. Who was Gregor Mendel, and why he's named as the father of genetic, and what he studied. So you can look at the history of Gregor Mendel, and you can see and focus on what he studied. He studied science, he studied mathematics, and his mathematics courses include the training in the new field of statistics. So Mendel knowledge of what later proved valuable in his research on heredity. So what helped Mendel in studying heredity was the statistic, the field of statistics, which is a part of mathematics. When Mendel returned to monastery, he taught in a high school. So he was a teacher in a high school, and also he kept a garden plot. So he has a garden in the monastery, and he started to planted seeds in it. And he's not studying only pea plant, but he studied also many other types of plants. One of these plants, which is important as he is remembered mostly for it, was his study on the pea plant or the garden pea. This is the common name for it. What is the scientific name for it? It's Pisum sativum. So what is Pisum sativum? It's the scientific name for the garden pea or the pea plant. Now, Gregor Mendel lived from 1822 to 1884, and what is important is this part. We say that Mendel's experiment with garden pea finally lead to what? Does it lead to the discovery of the DNA? No, it's not leading to the discovery of DNA. Does it lead to the discovery of the garden pea plant or the 7 character of pea plant? No, he observed them, he studied on them, but he's not discovered them. And so his the study on garden pea lead to what? It lead to the discovery of the basic principles of genetics. What is important here? He studied, and finally his study lead to the discovery of the basic principle of genetic. And that's why he named as the father of genetic. So if the question asked about who's the father of genetic, he was Mendel, or Gregor Mendel was the father of genetic. Genetic was found with the work of Mendel, especially on which plant, when he works on which plant, when he works on Pisum sativum. What is Pisum sativum? This is Pisum sativum. Pisum sativum, it's the pea plant, and you know that this pea plant, for example, we will talk about the traits in it. For example, we have this is pea plant, and you have purple flower. This is purple flower, and here we have the white flower. So if the question asks about this figure is for the pea plant, and here you can see the white flower and the purple flower. When we say white or purple, this is the phenotype of this plant, which means the appearance. This is white. We can say this is the phenotype of this plant. The phenotype of this plant is purple, which means the appearance of this plant is represented by wood. But if I asked about the genotype of this plant, we will talk about it later. It will be P capital, P capital, or P capital, P small for the purple flower. What about the white flower? This is the phenotype for it, represented by words. And if I say P small, this is the genotype of it, which represented by letters. We will explain that later. Now, what is the main difference between genetics and heredity? When we say genetics, it's a field of biology, so it's the science that study how the characteristics are transmitted from their parent to their offspring. But when we say heredity, it means the inheritance of the trait or inheritance of the character. So it's the transmission of the characters from their parent to their offspring. For example, you inherited your, let's say, skin color or your hair color or eye color from your parents. This is heredity. But when we say the science of biology that study the transmission of the character, this is named as genetic. Now, as you can see in this figure, we said this is the pea plant or Pisum sativum, purple flower and white flower. Now, what is the main difference between character and trait? And Mendel, when he studied the pea plant, he observed how many characters in the pea plant? He observed 7 characters in the pea plant. And when we see 7 characters, based on Mendel, he said that each character, we have 2 traits for it, 2 contrasting traits for it. So we can say we have 14 traits, or we can say 7 characters that he observed in the pea plant. What are these 7 characters and 14 traits that he observed? The characteristic that he observed, we said 7. Number 1 is the position of the flower along the stem. The position of the flower, if it's look like that, so this is named as axial. And if you can see it, it's on the top here. This is terminal. So we have axial and terminal. These are the traits for the flower position, or the position of the flower. The second character that he observed is the height of the plant. And according to the height, he said that we have 2 contrasting traits for the height of plants, which are tall and short. The third trait was its character, it's the pod appearance. And when we say pod appearance, we have 2 appearances for the pod. It's either like that, it's inflated, or constricted like that. So this is also the traits of the pod appearance. The other character that we have in the pea plant is the pod color. And for the color of the pod, you have either green or yellow. What about the texture of the seed? The texture of the seed, also this is a character, we have round and wrinkle. For the seed color, you can see that you have yellow and also you have green. What about for the flower color? For the flower color, there are purple and white. So according to that, we can say that Mendel studied how many characters? He studied 7 characters. Here these are the character. When we say character, it's the heritable feature that the plant inherited from the parents. And the heritable feature, for example, we say flower color, just flower color, or we can say, for example, seed color. So when I say them in general, I don't mention if, for example, the flower it's purple or white. This is a character. But when I mention it in specialist, for example, I say purple flower, I say white, I say green, yellow, or I mention the texture of the seed, if it's round or wrinkled. These are the traits, okay? So the traits are the variants of the character. They are the different variants of the character. But character, it's in generalist when I talk about any heritable feature that inherited from the parents to their offspring. So if we turn back to the definition of character and trait, you can see when we can say character, it's a heritable feature. So the feature or the character that the person, or we can say here the pea plant, inherited from where? From his parents. Like for example, flower color. You can see here, I don't mention if this flower is it purple or white. So at that time when I say just flower color, it's a character. But if I say flower, a purple flower, for example, or white flower, at that time this will be trait. The character determined by what? It's determined by gene. And we will talk about gene later. It's a segment on the DNA which is responsible for the character. What about the trait? For example, you can see when we said a flower color, we said that we have purple and white. So the purple and white are the traits. So what we mean by trait? Trait are the genetically determined variants, okay? Or we can say the 2 alternative forms based on Mendel. The 2 alternative forms of the character are named as what? Are named as traits. For example, when we say flower color, when we say purple or white, this is trait. And it's determined by what? It's determined by letters. These letters are named, we can say, alleles. For example, when we say gene, this is a gene responsible for example, the flower color. Both of them represent a gene. But when I mention only one letter or one of them, this one will be allele or factor. So trait determined by allele, while characteristics determined by gene. And as we said, based on Mendel's experiment, he found that every character, we said character is the feature that the pea plant inherited from the parents, has 2 contrasting traits. And as you see here in this figure, we see that every character, for example, if I talk now about the pot color, we can see that you have 2 contrasting traits for it, green and yellow. The same thing for the other 7 characters. For each character, you have 2 contrasting traits. This is only in Mendelian genetic. Please pay attention in the experiments of Mendel. So, for example, when we say flower color, we have purple and white. Now, according to Mendel's rule, each trait is represented by how many letters? When I mention a trait, for example, purple or white, purple flower, the purple flower represented by how many letters? Can we say only it's P capital or only it's P small? No, this is wrong. You should represent it by two letters. When I say purple, I wrote P capital, P small, for example, or P capital, P capital. This is purple flower. So we have two alleles or two letters. For example, also we have the white flower. For the white flower, we wrote also P small, P small. So also it's represented by two letters. So it means any trait that I want to write its genotype, I need to represent it by how many letters? By two letters. Two types of traits in the pea plant. We have the dominant and the recessive. The dominant, it can represented by two different genotypes. It can be either P capital, or we can write it as P capital, P small. For example, this is for the purple flower, which is a dominant trait. What about the recessive? The recessive trait, it's represented only by small letter. Both of them should be small letters, and like P small for the white flower. For the dominant traits, we can say the dominant parent can produce a recessive offspring. For example, if you have a dominant parent which is P capital P small and cross it with P capital P small, so at that time you can see it can produce P small, which is a recessive. So we can say the dominant parent can produce recessive, but the recessive parents never can produce dominant offsprings. Now, what do we mean by genotype and phenotype? The genotype is the genetic makeup of the organism, and we represent it by letters, and it consists of the alleles. And we know that each letter represents an allele. For example, when you say P small, A capital, A capital, A capital, A small, all of these are letters. So all the letters represent what represent the genotype of the organism, which is the genetic makeup of the organism. For example, when I want to write for purple flower, I say P capital or P capital P small. But for the white flower, it's P small, P small. This is the genotype of the organism. And the genotype, we have two types of genotype. The first one is named as the homozygous or the pure one, and the second one is heterozygous or the hybrid. And when we say homo or pure, it means we will use the letters or the alleles, they should be like each other. Like for example, when we say A capital, so they are similar to each other. Both of them are capital letter, or both of them can be small letter. So these will be pure or homozygous. So we can say the pure or homozygous, we will divide it also into two types. One of them it's a homo. We can say this is the homo dominant. It's made up of two letters, both of them are capital. Or we have homo recessive, both of them are small letter. But for the heterozygous or the hybrid, we can say we have only one type, which is always its dominant, represent the dominant. For example, when we say P capital P small, A capital A small, T capital T small, all of these are made up of capital and small letter. And if it's made up of both capital and small, we say it's a hybrid or hetero. So, for example, this P capital P small represent the purple flower, and we know that purple is dominant. A capital A small, it's for axial, and it's also dominant. T capital T small, it's tall, and it's also dominant. But all of these are hetero, or we can say hybrid, because they have different we can say different letters in one of them. It's for example the letters, one of them is a capital letter and the other one is small letter. That's why we say it's hetero or hybrid. Okay. Now, if we talk about the phenotype, on the other hand, the phenotype, it's not the genetic makeup, it's the appearance of the organism. What you can see, for example, when we say purple flower, this is the appearance of the flower color. And when we say, for example, inflated pod, all of these are appearance and they are represented by letters or words, they are represented by words. Okay. For example, if he asks about the phenotype of P capital or P capital P small, it will be purple flower. while the phenotype of p small, it will be the white flower. Okay, and we have an important note here that a phenotype does not always indicate the genotype. This is very important, and this is only in Mendelian genetic, which means incomplete dominance. We will talk about complete dominance, and it's a type of Mendelian genetic. In Mendelian genetic, we can't say that usually that the phenotype represents geno. Why? Because we have two reasons for that. The first reason is that the recessive alleles, because if the recessive allele comes with the dominant one, at that time the dominant will cover or mask the effect of the recessive. So it will not appear by the recessive trait, instead it will appear by the dominant trait, which is purple. And that's why the phenotype does not always represent genotype. Why, like for example, when we say P capital P small, this is recessive, but it's show the dominant trait here because of the recessive allele. When it comes with the dominant allele, the dominant will cover the effect of it. The second reason is that because of the certain environmental factor. Do these certain environmental factors they affect the genotype or phenotype? They affect the phenotype. That's why the phenotype does not always represent the genotype. Why? Because sometimes these factors can affect the phenotype of the organism. For example, if you have in the soil lack of proper nutrition, lack of water, lack of, for example, the exposure to sunlight, it's not enough. At that time, what will happen? The genetically tall plant may remain short, and that's why we can't say always the phenotype represent the genotype in the pea plant. This is only for Mendelian genetic. While from the genotype, yes, we can always indicate or represent the phenotype. Why? For example, if you have the genotype P capital P small, you directly will know that this plant is purple. Or for example, if you have P small, you will directly know that the phenotype of this organism or of this genotype, it's white flower. So from geno, we can know the phenotype, but from phenotype, we can't always represent the genotype of it. Okay, now if we talk about the Mendelian trait, as we said, they named as complete dominance. And why the Mendelian traits of when we study the pea plant, it's named as complete dominance? Because if you have recessive allele with the dominant one, the dominant will completely cover or mask the effect of the recessive. That's why when we write, for example, P capital P small, we say it's purple. We can't say it's white, okay? Even though it has the recessive allele in it. So that's why it's named as complete dominance. And in complete dominance, we said that phenotype, it's not always indicate the genotype. While in the other types of non-Mendelian genetic, that we will explain it in the second section, we can say non-Mendelian traits like incomplete dominance and codominance. Here, the phenotype, it's always indicate the genotype, and we will talk about it later in section 2. According to Mendel genetic, each trait is represented by, pay attention, it's two letters. So each trait, for example, when we say that seed color, if I talk about seed color, this is a character, right? But if we say the yellow seed color, this is a trait. And this trait, it's represented by how many letters? It should be represented by two letters, okay? So we have Y capital, for example, okay? Or maybe Y capital, Y small. This is for the dominant trait. And what about if the trait is recessive? So at that time, it will be represented by also two letters, but with lowercase letters. Like for example, we can say for the green seed, we were represented by Y small, Y small. Okay. When we write it, we write the first letter. We use the first letter of what? We use the first letter of the dominant trait, not of the recessive trait. For example, when I want to mention the green seed, I don't write it as GG or G small. I wrote it as what I said that for the seed color, the yellow is the dominant one and the green is the recessive. So the recessive will be Y small, Y small. What do we mean by that? We mean that, as we said before, we have seven characters of the pea plant. We have the seed shape, seed color, pot color, flower color, the location or the position of the flower, and the size of the plant. These are the characteristic. I talk about them in general. We said these are the heritable features. And for each character, we have two contrasting traits for it, right? We have the dominant one and the recessive one. So for the seed shape, we have round is the dominant while the wrinkle is recessive. How can I write them? The round now and the wrinkle, they are the phenotype. Why? Because we said that phenotype is the appearance of the organism and it represents by words. What about if I want to write the genotype of it? The genotype, we said it represents by letters. And these letters for the dominant, we said we have two types. Either homo, which means pure, and will be R capital for the round, and maybe hetero or hybrid and can be R capital R small because it's dominant, and the dominant represented by two genotypes, either homo or hetero. What about wrinkle? Wrinkle is recessive. How can I write the wrinkle? Is it WW, or I should use the first letter of the dominant and make it as small? Yes, this is the rule of Mendel. We will use the first letter from the dominant and make it in small letter or lower cases. So we say for the wrinkle should be R small, R small. And pay attention, for the recessive you have only one genotype, which is small letter, small letter. Now if we talk about the seed color, the dominant, it's yellow. And for the yellow, because it's dominant, and we said for the dominant we have two types, homo or hetero, so it will be either Y capital or Y small, Y small. What about the green color? The green color is recessive. At that time, we can write it as Y small, Y small. The inflated pod, it's a dominant trait, and this dominant trait, which means the appearance of this cover of the plant, of the pea plant, because it's dominant, we have two genotype for it. It will be either I capital or I capital I small, homo or hetero. What about constricted? I can't write it as CC. We should write it as the first letter of the dominant one. So it will be small I small I small. The same thing for the green pot. Pay attention for the seed color and the pot color. They are the opposite of each other. For the seed, the yellow is dominant. For the pot cover, which is, this is the outside cover of it, the green is dominant. So at that time will be G capital or G capital G small. What about the yellow pot? At that time will be G small because it's recessive and have only one genotype. Purple, as you know, it's the dominant flower color. We have P capital or P capital P small. What about the white? Will be P small, P small. The axial flower, the same thing, it's dominant, so A capital A small or A capital A capital. What about terminal? It will be A small because it's recessive. For the tallness of the plant, we have T capital, T capital, T small. And for the short plant, we can represent it by T small, T small. This table is very important, and you should know that you have a seven characteristic for the pea plant, and that's why we say that pea plant has how many chromosomes? It has 7 pairs of chromosomes. What about the gametes of the pea plant, like the pollen grain? The pollen grain has only 7 chromosomes in it. So for the dominant trait, you can write it as you have two types, two genotypes. Either will be homo or pure, or can be hetero or hybrid. While for the recessive trait, they only represent by one type of genotype, which is small letter, small letter. Okay. Now, for every scientist, you should know the observation of the scientist, the experiment that he did, and the result with conclusion of his study. The observations of Mendel. First, he observed what? He observed when he planted the seeds for the flower color. He observed that the purple flower plants, they grew from most of the seeds obtained from purple. So when he planted seeds of purple flower, he find that all the mostly we can say most of the flowers that are produced are purple, but some of them they grow to white flower. Why that happened? He mentioned that later, that something happened and something control the crosses that happen, and this has happened because the seeds may have heterozygous genotype. That's why they can produce the white color. And for the plant height, the same thing happened. He observed that while the tall plants grew from most of the seeds obtained from the tall plant, but some of them can grow to short plants from the tall plant's seed. So he want to find an explanation for these variations or for these differences that he can find in his experiment. So what he observed, for example, let's say for when he grow the seeds of purple plant, they mostly grow to purple, but some of them they grow to white plant, while a white flower plant. And for the height of the plant, the same thing happened. When he planted the seeds for it, mostly they grow to tall plant, but some of them grow to short plant. He want to find explanation for these variations that he saw. So how was Mendel able to observe how these traits were passed from generation to the next generation? How? By controlling how the pea plants were pollinated. What do you mean by pollination? Pollination is the transferring of pollen grain. Pay attention, not the transfer of anther or the stigma. It's the transfer of the pollen grains. These pollen grains are produced where? They are inside here. They are inside the anther or the male part of the plant. They transfer from the male part to the female part, which is named as stigma. So the pollination, transferring the pollen grain, they transfer from male to female part, which both of them are on the same flower. The male part is named as anther, while the female part is named as stigma. The anther is the male reproductive part of the flower and produce the pollen grain in it, while the stigma is the female reproductive part and contain the ovary. So this pollen grain will transfer from the anther or the male part to the stigma, which is the female part. We have two types of pollination. We have self-pollination and cross-pollination. If I talk about the self-pollination, which is normally happen in the pea plant, we can say we have two types of it. The self-pollination happen when the pollen grain transfer from where to where? From the anther to the stigma. We can say transfer from the anther of this flower to the stigma of the same flower, or transferring of the pollen grain from anther of a flower to the stigma of another flower, but on the same plant. Okay, this is for the self-pollination, and the pea plant normally reproduce by self-pollination. The other type of pollination that Mendel did it manually, it's named as cross-pollination. In cross-pollination, the pollen grain will transfer from anther of a flower to the stigma of another flower on another plant, which is then genetically different. Okay, this is the cross-pollination, and we said that normally the pea plant reproduced by which one? It reproduced by self-pollination. How we know that it's a self-pollination if they are genetically identical? For example, T small, when you cross it with another short plant, which is also T small, this is a self-pollination. T capital, tall plant with another tall plant, and both of them are with the same genotype. So they are genetically identical, so we said it's also self-pollination. And also for T capital T small with T capital T small, they are the same, so it's also self-pollination. While for cross-pollination, they should be genetically different from each other, like when you cross tall with short and they are completely different in their genotype, or T capital, which is for tall, and another tall but with genetically different. And here also we have the short with a tall and they are not identical, so at that time we say this is a type of cross-pollination. Okay. For example, if we want to produce a tall pea plant can be produced by both of them, self-pollination and cross-pollination. In self-pollination, yes, you can produce the tall plant, and also by cross-pollination, we can produce the tall pea plant. Now, the Mendel methods of pollination. We know that pea plant, we said it's normally reproduced by which one? It's reproduced by self-pollination. And we said that self-pollination, it's the transferring of the pollen grain from the male part, which is the anther, to the female part, either on the same flower or another flower, but on the same plant. They should be genetically identical. For example, if you cross A capital with A capital, this is self. A capital A small with A capital A small. This is also self-pollination. A small with A small. If you cross the trait by itself, so it's self-pollination. Self-pollination, how it can be prevented? We can prevent self-pollination by removing what? Very important. By removing all the anthers, the male parts from the flowers of a plant. So we can remove all the anthers. At that time, the plant will be antherless, which means it consists only of the female part. Then we do the cross-pollination manually as what Mendel did. And it can be performed manually. How? By transferring the pollen or the pollen grain from the flowers of second plant. Which plant? The plant that have the anther, to the stigma of the antherless plant, the plant that do not have the anther. Okay, so normally the plant reproduces by self, but we can prevent it how? By remove all of the anthers. From where? From the flowers of a plant. Okay. Now, by preventing, why Mendel did that? Why he did prevent the self-pollination and he did cross-pollination? Why he did that? In order to, number one, to be able to select the parents as he want and to observe the traits that appear in the offsprings. This figure is also important figure. If we go to talk about what Mendel did, we said he removed the anthers from flower of the plant. So which one here the anther is removed? This one, the anther. These are the anthers. Okay, you can see it. These are the anthers, which is the male part. And here we have the stigma, the female part. So this flower, let's say it's A, or the white flower. Now it has both male and female part, which is the anther and the stigma. While the figure B, or the purple flower, it has only the female part and there is no male part. We can say it's antherless. It means no male part. It only has the female part. And what he did cross-pollination. So if the question asked about here, what happened here? It's cross-pollination. And you know in cross-pollination, the pollen grain will transfer from anther to the stigma. And which one has the anther? This one has anther. So we will transfer the pollen grain from here, from the plant that has anther, to the stigma of the antherless plant, the plant that do not has the anther. And this happened manually. The Mendel did it manually, and he transferred the pollen grain from a flower to another flower, which is antherless and has no anther in it. So if the question asks about how Mendel was able to control the breeding or the crossing that happened, is it by cross or self-pollination? He was able to control it by cross-pollination, by transferring the pollen from, he did it manually, and he transferred the pollen grains or the pollen from where? From the anthers of one plant. Which plant here? From the white plant toward what? Toward the purple plant, which is antherless. So we can say he removed the anther. Is it from the purple or from the white? He removed the anthers from the purple flower. And he transferred the pollen grain from the white to the purple. Very important. So when he started his experiment, he initially, or at first, what he did, he studied each characteristic, and its contrasting traits. What do you mean by that? For example, he studied the flower color with both contrasting trait for it, which is purple and white. And he studied, for example, the for the height of the plant, which is a character, and for both of them, tall and short. So when he studied each character, we have 7 characters in pea plant, and each character with its contrasting trait, it means you have 14 traits. But what he did first, he grown the plants that were true breeding for each trait. What do you mean by true breeding for each trait? True breeding, it's also, we can say it's homo or pure plants. Those plants that always produce offspring with the same trait when they self-pollinate. Very important. So we can obtain true breeding by self-pollination, but for several time or for several generations. For example, we can say T capital T capital. This is true breeding. It's pure, it's homozygous, and he did it for many generations in order to finally obtain all the plants are T capital, and they are pure. If we cross it many times, they only produce the same traits of the parents. This happened by self-pollination for several times or for several generations. And the same thing for example, T small, T small. This is also pure or true breeding plant. So how he get the true breeding? By self-pollination for several times in order to exclude all the recessive traits or all the recessive alleles, and finally he obtained it as pure or homo or true breeding. Okay, how he get it? Is it by self or cross-pollination? It's by self-pollination. Now, for example, let's say the pea plants that are true breeding for the trait of yellow pod. You know that yellow pod is recessive because the cover, we said the green pod is dominant. So the yellow pod that is recessive self-pollinate to produce the offspring that have yellow pod. What do you mean by that? G small, this is for yellow pod, crossed by G small, they only produce G small. So this is the definition of true breeding. They produce offspring should be the same trait of the parents, okay? And it's happened by what? By self-pollination. For example, we have a question here. Mendel produced repeating plants. How? By self-pollination of the pea plant for several time or several generation. He finally obtained or get how many true breeding type? He get 14 true breeding plant, and for each of the trait he observed, he get one. Now Mendel finally he get, as we said before, 14 true breeding plant type for all the traits that he studied, one for each of the trait. Now we will explain the experiment on the figure, and we'll go to see the P generation, the F1 and F2 on the figure, and then we'll come back to explain it. So we have three steps. These are the experiment of Mendel. It has three steps, and these steps are very important. This figure is very important for ministry exam. In this figure, and what Mendel did, he did self-pollination for several generations, as we said, to prepare the true breeding. Why he did that? To get the parent generation, or to select the parents, which is the P generation. So in the first step, he produced a true breeding P generation. And P generation, or the parent generation, we have P capital. This is for all these purple flower plant, and we have P small for all these white flower plants. This is the first type of Mendel. So how he get P generation? He get the P generation how? By self-pollination for several generation. Now, how he get in the second step, how he get the F1 generation? F1 generation, or which is named as first filial generation, he get it by cross-pollination of whom? Cross-pollination of P generation. Okay, so the F1, as you can see here, they are all purple and they are all P capital P small. Why they are P capital P small? Because if you cross the purple with the white flower, and you know the purple here, it's all P capital P capital. We will talk about the Punnett sequel later, and the white flower, it's P small P small. So when you cross them, all of the offsprings will be P capital P small, or heterozygous, or we can say hybrid. The F1 generation produced by the cross pollination of the parent, or the P generation. Can we say they are produced by cross pollination of parents with two contrasting traits or with two similar traits? It's with two contrasting traits because one of them will be purple and one of them will be white. But what do you mean by cross pollination? As we said before, when he did cross pollination, he did it manually. How? By removing all the anthers of the dominant plant and transfer the pollen grain from the white flower to the purple flower. This is what he did in order to make the cross-pollination and to produce the F1 generation. Now, in the third step, he let the F1 generation to cross themselves by self-pollination. So he did self-pollination for all the first or F1 generation, and what he get in F2, they named as a second filial generation. He get one of them would be purple P capital P capital, two of them are P capital P small, and one of them is P small P small. So we say three purple and one white, while in F1, as you can see here, all of them are white. We can say as ratio 100% they are purple, while here 75% they are purple and 25% it's white. Okay, so how he get F2? He get F2 by self-pollination of what? Of F1 generation. How he get F1? By cross-pollination of P generation. How he get P? By self-pollination for several generation. So you can, for example, in order to not forget in the exam, you can make it as easiest way by writing, like we can say this is self-pollination for several self-pollination to get the P generation. Then in order to get F1, he did cross, and again by self, he get what? He get the F2. So you have two times you can say in the first step and in the third step we have self-pollination, while in the second step we have cross-pollination. So it's self, cross, self. In order to get P generation, he did self. In order to get F1, he did cross-pollination. And in order to get F2, he did self-pollination of F1. Or we can say he get the F2 by self-pollination of parents that have the same trait. Okay, so returning back to the definitions of them, for example, we say the P generation or the parent generation are the first two individuals of the true breeding pea plants. And how he get them? Is it by self or cross? It's by self-pollination. And as we said, they are true breeding for contrasting trait because, as we said, we have purple and white. Now, when he did cross-pollination for both these parents, he get what? He get F1 generation. And after that, he get F2 generation. So F1 generation are those offspring that come from where? From the P generation by cross-pollination of the P generation. What about F2? He get F2 from where? From F1. But how? By self-pollination of the F1 generation. So Mendel called the plants in this generation as second filial generation or F2 generation. They are the offspring that come from self-pollination of F1 generation. Or we can say from self-pollination of parents that have same traits because it's we cross purple with purple, P capital P small with P capital P small. And that's why it's appeared by, as we said, P capital P small. If we cross it with P capital P small, at that time you can see what? You can see that you will get how many different phenotype. One of them will be P capital, which is, as you know, is purple. And two of them will be P capital P small, which are, we can say, also purple. And one of them will be P small, which is white. So that's why we say three of them white, purple, and one of them it's white. Okay. And Mendel did the same thing, and all he get the true breeding for all these 7 character, and he repeat the experiment for all of these traits. We have contrasting trait. We said that we have 14 contrasting traits, as we said before, and he did it manually. He cross pollination manually in order to get F1 generation, and also he select the parents by doing true breeding and by doing the self pollination. And after that he did for all these 7 character, he studied each one individually with its contrasting trait. He get these results, and these results you can see that every time he did the cross, you can see that in F1 generation, first filial generation, only one of the parent trait will appear. Which one will appear? Is it the dominant or the recessive? The one that appeared, he named it later as dominant trait because it's appeared from first generation in F1 generation and in a percent of 100%. So one of the parent trait will appear and this one will appear, which is the dominant trait, which is here, for example, for the position of flower, it's axial. For the tallness of the plant, for example, if he cross tall with short, he find that all F1 will be tall. So this is the dominant. And for the F1 of the pod appearance, in the F1, all of them will be inflated, which is also the dominant. For the color of the pod, we said the dominant is the green, so all of them will be green in the F1 generation. And for the seed, also for the texture of the seed, we have round and wrinkle, and all of it will be round in the first generation. So for all these other character or the traits, he did the same, and the one that appear in F1 is the dominant trait, which appeared by a ratio of 100%, or we can say 1-0. Now in F2 generation, he find the same result also. He saw that all the ratios are nearly equal to 3-1, which means three of them will be dominant and one of them will be with the recessive trait. So this is the observable ratio. For example, 3-14-1 and 2-84-1. These are observable ratio, and the predicted ratio at that time we say it's 3-1. Okay. So the predicted ratio in F2 generation is 3-1. This is for the phenotype, which means, for example, we can see 3 purple, 1 white, or 3 tall, 1 short. Okay. So we have 7 character for a Mendel experiment or for the bee plant. You have pod color, pod appearance, seed color, seed texture, flower color, height of the plant, and the position of flower. For every character in Mendelian genetic or in Mendelian experiment, they are dominant and they are two traits, contrasting traits. For example, for the pod color, we have green and yellow. For the pod appearance, inflated and constricted, and for all the other the same thing. For each one of the character we have two contrasting trait. This is only for pea plant or for Mendelian genetic. And what he did in his experiment, as we said, he get the P generation, F1 and F2 generation. P generation, we have one dominant green and one recessive yellow. So he crossed the contrasting traits in the P generation. We have these contrasting traits. He get the true breeding for this plant by self-pollination. And then by cross-pollination of P generation, he get F1 generation. In F1 generation, he saw that all the offsprings here or all the first filial generation, and for all these characteristics, he found that only one of the traits of the parent will appear. Which one will be appear? It's only the dominant will be appear in the F1 generation, and all the dominants here are hetero or hybrid. Now after that, he did the self-pollination again of F1 generation to get F2. When he get F2 from F1 by self-pollination, he saw these results. All of them are 3-1. 3 dominant and 1 recessive will appear. So if we want to compare the P generation with F1 and F2 generation, in P generation, as we said before, both parents are homozygous. One of them would be the homodominant and the other one will be homorecessive. And in F1 generation, we said that all them are dominant and they are heterozygous. What about in F2 generation? In F2 generation, we can say 50% are homo and 50% are hetero. Why? Because we know that one of them, we said it will be P capital, P capital. Two of them will be P capital, P small, and one of them will be P small, P small. So this one with this one, both of them will be homo, and these two are hetero. So we can say 50% are homo and 50% are hetero or hybrid. Here in P generation, we said one of the parent will be capital letter because it's homo dominant, while the other one will be recessive and will be in small letters. While in F1 generation, we said all of them are hetero. And we can see in F2 generation, 25% they are homo dominant and 50% are hetero, where 25% are homo recessive. The genotypic and phenotypic ratio is 1-0. What do you mean by that? We said that in F1, all of them are hetero and they are dominant. So when we say all of them, it means 100%. 100% as ratio, we can write it like that. We can say 1 to 0, it means 100% the dominant trait will appear, and 0% the recessive will appear. This is, we can say for both of them, for geno and pheno. It means for genotype, when we represent it by letters, like I say, all of them are P capital P small. This is the genotype. And for the phenotype, we can say it's the appearance. So all of them are purple, for example. That's why it's 1, 0. What about in F2 generation? In F2, the genotypic ratio, when we represent it by letter, we say it's 1 to 1. One of them is P capital, P capital, two of them P capital, T small, and one of them is P small, P small. While for the phenotypic ratio, it's very important to know the phenotypic and genotypic. The phenotypic is the appearance, and what we see is that 3 purple and 1 white. Okay. The P generation are true breeding, and what we mean by true breeding are those plants that whenever you cross them by self-pollination, they produce the same traits of the parents, and he get them how by self-pollination for several generations. While F1, they are not true breeding, and for the F2 generation, 2 of them are true because they are homo, and 2 of them are not true breeding because they are P capital P small. Okay. We said that P generation are produced by self-pollination, okay, and the F1 it's by cross-pollination of the P generation, or we can say cross-pollination of 2 contrasting traits, which are the P generation. The F2 we get it from where? From self-pollination of F1 generation, or we can say by self-pollination of parents, which is F1, that have the same traits. In another experiment that Mendel did, for example, he crossed a green pod pea plant with yellow pod pea plant, and we know that the green pod is dominant, which is the cover of the plants of the pea plant seed, and the green is dominant. And he crossed true breeding green pod, which is G capital G capital. Why it's G capital G capital? Because it's true breeding, it's homo, it's pure. He crossed it with the contrasting trait for it, which is yellow pod, the recessive one, which is G small, G small. And why G small, G small? Because true breeding and recessive. So if we cross G capital with G small, G small, what will happen is that in the F1 generation, you can see that all of the F1 generation will be G capital, G small. So F1 generation, all of the plants will be G capital, G small, and we know G capital G small, it's green, it's dominant. So we can see when he did the how he get the F1 generation by the cross pollination of what? Of the P generation. The P generation, as we said, they are G capital with G small, G small. How he did the cross pollination? You know that he did it manually by removing what? By removing all of the anthers. He removed the anthers from where? From the dominant plant, so which means from the green. And then he transferred the pollen grain from the plant that has anther, which is the yellow, to the green. So again, he did the cross-pollination. How? Manually. How? By removing all of the anthers of the green pod, which is the dominant, and then transfer the pollen grain from the anther of the yellow pod to the stigma of the green pod. After that, he found that as a result, the F1 generation, all of them, they grew to green pod, which is G capital G small. We can say 100% or 1-0, they are G capital G small. This is the genotypic ratio. And 1-0, they are which color? They are green color, which is the dominant. So we can say in the F1, only one of the parent trait or one of the parents will appear, which is the green one, the dominant one. Now, how he get the F2? We know that F1 by cross-pollination. How he get F2 generation? By self-pollination. He did self-pollination of F1, which means he crossed these by themselves. He crossed G capital G small by G capital G small. What will happen is that the offspring that will be produced when you cross G capital G small with G capital G small, one of them will be G capital green, homo, and 2 of them will be G capital G small. As you can see here, they are also green, and one of them will be what will be G small, which is the yellow pod. So you can see in the F2 generation that he get them by self-pollination of F1 generation, or we can say he get F2 by self-pollination of parents that have the same traits. As you can see here, they are green. So the result of it, it's three, we can say 3 to 1, which means 3/4 of them are green pod and 1/4 it's yellow pod. So from the 4, we have 3 of them are green and 1 of them is yellow, which is the recessive one. So in every cross that Mendel did, he found that in F2 generation, the dominant will appear in 75% and 25% the recessive trait will appear. And if we go to talk about the genotype, not the phenotype, we can say we have one G capital, G capital, two of them are G capital, G small, and one of them is G small, G small. So the ratio for genotype, it's 1 to 2 to 1, while for phenotype, it's 3 to 1. In every cross he did, he get the same result. He get that in F1, 100% the dominant trait will appear, and in F2 that the dominant will appear in 75%, while the recessive appear in 25%.